Hey students, I wanted to create just a quick video of the mini lesson that we did this morning, like I mentioned. And so again, if you missed any of the notes, take a look at this video. So what I started this morning talking about was about the endomembrane system. And I showed you this particular slide where the endomembrane system is composed of several different organelles in eukaryotic cells working together in a system. And those organelles I mentioned were the nucleus, the smooth and rough endoplasmic reticulum, the Golgi complex or the apparatus, and lysosomes. What you also have involved in this system are things called vesicles. Vesicles which are responsible for transporting substances from one portion of the endomembrane system to another. Okay, so those are the components of the endomembrane system. What does the endomembrane system do? What is it responsible for? Well, the primary thing that it's responsible for is first, starting in the nucleus, a gene is transcribed into mRNA, and that mRNA makes its way to the rough endoplasmic reticulum, right? And that rough endoplasmic reticulum has ribosomes on it, which allows for that mRNA to be translated into amino acids and into a protein. Now, those ribosomes are embedded in the membrane of the rough endoplasmic reticulum. And since that's the case, what happens is a vesicle actually encapsulates the newly translated protein into a structure that looks like this. So this is what a vesicle looks like. It's composed of phospholipids. Remember that this is spherical as well. So it's like this spherical pod that encapsulates the newly translated protein and moves it from the rough ER to the Golgi, like we see right here. Right? So we have transcription that occurred in the nucleus, translation that occurs in the ribosome, and then a vesicle comes off of the membrane and moves towards the Golgi. When it gets to the Golgi, remember that there are enzymes and different processes that happen in the Golgi that help modify proteins, whether that is adding or removing different elements from its structure, helping it finish folding. But once it's ready, that vesicle, which entered the Golgi on this side by actually fusing, the membranes fuse with one another, that protein makes its way to the other side of the Golgi, actually through vesicles going sequentially through the different sacs of the Golgi. And when it gets to the other side, there are really three main places where that modified protein can go. The first option is that the vesicle that comes off of this side of the Golgi can sometimes contain multiple enzymes, specifically hydrolytic enzymes, and that vesicle can mature and become a lysosome, right? We know lysosomes, that's an organelle, a membrane-bound organelle that contains hydrolytic enzymes that are responsible for breaking down macromolecules in the cell. And so that's one option. The second option is that the modified protein can be sent in a vesicle to the outer plasma membrane, where, like we see in this figure, that vesicle fuses with the membrane and releases its contents outside of the cell. That's called secretion. So it can either become a lysosome, it can be secreted, or the other option is that it can combine with other vesicles to form a little bit of a larger storage vacuole, for example, to be used later. So those are three major pathways that this endomembrane system ends up leading to: secretion, lysosome, or storage by combining it in other vesicles. Now, one thing to note here, right, is that vesicles are made up of these phospholipids. And if we have constant vesicles coming off of the uh, endoplasmic reticulum, right, it makes sense to think about, well, won't eventually the rough endoplasmic reticulum run out of phospholipids? Um, and while it's a great question, the smooth ER you read actually produces more lipids. And so it gets replenishes as those vesicles pinch off and go towards the next locations. Okay, so that's the endomembrane system. Okay, so that's the endomembrane system. The last part of this was I mentioned for the question of how are vesicles transported from one location of the cell to the next, or one portion of the endomembrane system to the next. And that gets down to the cytoskeleton. So if you remember the cytoskeleton, there's three different types of fibers that make up the cytoskeleton. With regards to vesicles, the microtubules, which are labeled right here in this left-hand photo, they make up this very dense network on which vesicles are physically pulled along from one place to another. So there are these specific type of proteins that actually walk along microtubules, like we see right here, while also holding a vesicle. And so let's say this vesicle had come off of the rough endoplasmic reticulum. There's a microtubule on which a motor protein would walk that vesicle to the Golgi, right? And from there, wherever it needs to go afterwards. So just a quick note, that's your answer for the third question here. Vesicles are transported by motor proteins walking the vesicle along microtubules. Okay, the other thing that I talked about this morning had to do with ribosomes. So just a quick note on ribosomes. So we know that ribosomes are involved in the translation of mRNA into amino acids, which a chain of amino acids folds into a protein. But what I want to make clear here is that ribosomes in eukaryotic cells can be found in two locations. So the two locations where they can be found are one, in the rough endoplasmic reticulum. They're embedded on the membrane of the rough ER, and they can be found in the cytoplasm, which is the liquid of the internal environment of the cell. So we see ribosomes here and ribosomes here. Now, what is the function of free ribosomes? By free ribosomes, I mean the ones that are not attached to the rough ER, so these ones. The function is that normally any proteins that are made by free ribosomes operate in the cytoplasm. So we're going to talk about in Unit 3 a bunch of different reactions that happen inside of a cell. A lot of them happen in the cytoplasm. And so it makes sense that a protein that's made by the ribosome is going to stay in the cytoplasm if it's made by a free ribosome, as opposed to ribosomes found on the membrane of the rough ER, right? That's part of the endomembrane system that we just talked about. And normally those proteins are going to end up either being secreted out of the cell, stored in a lysosome, or stored in a vacuole, right? And so those are not necessarily just going to be used in the cytoplasm. They're going elsewhere. So that's just a quick note about ribosomes. Remember as well that ribosomes, they don't have membranes around them, right? They are not membrane-bound organelles in the sense that they are not surrounded by a double phospholipid bilayer. Okay. Okay, the last thing that I talked about had to do with cell size and diffusion, right? And diffusion is simply the movement of substances from an area where there's a high amount of that substance to an area where there's a low amount of that substance. Now, the first part of this, I talked about why a cell needs to be able to efficiently exchange materials with its external environment, or in other words, diffuse substances with its external environment. And the major reason is that there are nutrients and waste products that need to be taken in and out of the cell regularly. So one of the most basic components that a cell needs to either get rid of or take in is carbon dioxide and oxygen, right? So there needs to be a very efficient exchange of those two gases across the cell membrane. And so a cell needs to be able to do that efficiently in order to survive. Other substances like glucose need to not only be able to make their way into the cell, but they also need to be able to make their way throughout the cell once they're in there. Right? There has to be an efficiency of getting that glucose to where it needs to go. And this boils down to how big a cell is. We can think about the efficiency of a cell to exchange materials with its environment if we think about this ratio of its surface area to its volume. And for your second question in your notebook, how can we predict how efficient the cell is in exchanging materials with its environment? The key takeaway is that a cell with a higher surface area to volume ratio will be more efficient in exchanging materials with the environment than a cell that has a lower surface area to volume ratio. So I walked through this figure right here, which is showing a model of a cell, which in this case is a cube, with a side length of 1. If you were to calculate the surface area, it would be 6. The volume, the space inside, would be 1. And so the surface area to volume ratio, which is just surface area divided by volume, would be 6. However, if you increase the volume such that now you have a 5 by 5 cube, right now your surface area is 150, your volume is 125, and look at your ratio, right? Your ratio has gone down to 1.2, and so it's not as efficient as the cube with the side length of 1. However, if you modify that even more, such that now you have a cube that's composed of a bunch of other smaller cubes, then you haven't increased the volume, but you've increased the surface area, because now you have in the spaces in between the cubes, that's now surface area because it's exposed to the outside. So now you've increased your surface area volume ratio to 6 again. So this cube or cell would be more efficient than this one in exchanging materials. And the same thing is true for a sphere, right? Most animal cells are spherical in shape, or at least approximately spherical. And so you can think about, if you change the diameter of that cell, you can think about the surface area to volume ratio, and the equations are right here. As you increase the diameter, that surface area to volume ratio goes down, and so the smaller cells are going to be more efficient in exchanging materials. This also explains why cells are small to begin with. Okay, so the last part of this was, I talked about there are some cells in the body that, because of their function, they have specific structures that allow them to increase their surface area to volume ratio drastically to make them extra efficient in exchanging materials. And so I had us think about what are some areas of the body that require that kind of efficiency. And the two major ones are in the intestines, and I also mentioned the neurons. Right? For the intestines, if you think about in your intestines, one of the major jobs is it has to absorb your digested nutrients. And to accomplish a very high surface area to volume ratio, these intestinal cells, they have these structures called microvilli, which are these protrusions that allow for high surface area, but not drastically increasing the volume, so we get a high efficiency in exchanging materials with the environment, which in this case is absorbing from the inside of the small intestine. Okay. The other example is a neuron. Right? A neuron, if you look at the two ends, has all of these different protrusions and folds that allow for efficient exchange of neurotransmitters with other neurons. And we'll talk about that in our cell communication unit. But these two are big examples of increasing the surface area to volume ratio to allow for very efficient exchange with the environment. The last example that I had you think about was in your lungs, your alveoli cells, the interior of your lungs, where cells are involved in that gas exchange of carbon dioxide and oxygen with the environment, right? Those cells, they have very similar structures to what we see in the intestines because they also have to be very efficient in exchanging materials with the environment. Okay, now to wrap this up, I skipped over this, but I did mention this in class. In the AP exam and in my class, if you're ever asked about, predict which cell is going to be more efficient, they'll likely have you calculate a surface area to volume ratio. You won't be expected to memorize surface area and volume equations. You'll always be given this on my exams and the AP exams. So you've got things like the volume of a sphere, the surface area of a cube, all of those things that you might have forgotten about from geometry will be given to you. Okay, so I just went over all that I went over in my mini lesson this morning. I hope that was helpful, and please reach out if you have any questions for me. Have a good rest of your afternoon.